Okay, thank you. Okay, so I'm Saori Tanaka, a computational neuroscientist in Japan. So at first, thank you for giving me such a great opportunity to introduce our project in such a great event. Okay, so, Today, so I'd like to introduce our research project of human neuroimaging database focusing on psychiatry and neurological disorders in Japan. Okay, so my talk starts from AI because this session's theme is AI. Okay, so as you know, AI is now very popular and become a great tool in academic situation. So in neuroscience, AI enables us to identify hidden features in vast neural dataset, supporting insight into brain function and the disorders. Okay, so in particular, recent success of machine learning techniques have been remarkable in the neuroimaging research for psychiatric disorders. So I show an example in classification on psychiatric disorders based on brain imaging data. Okay, so we collect data from healthy individuals and patients and input these data into machine learning algorithms. And the AI creates a classifier based on the characteristics of the disease. And the new data is applied to the classifier to help diagnose the disease. But we face a big problem. It is the amount of data we need. So recent study demonstrated that for producing replicable findings and reliable findings, machine learning classifier should learn from huge MRI dataset, identically 1,000 to 10,000 or even more. So it is meaning the enrollment of large numbers of patients. It is not realistic to recruit such many patients with psychiatric disorders or neurological disorders at a single clinical site. Additionally, it has been difficult that neuroimaging markers, classifiers of psychiatric disorders built from a single database can be generalized to data measured at different sites. So therefore, a large scale and multiple site imaging dataset in collaboration among multiple imaging center, imaging site is required. Okay, so I'd like to introduce our preceding project, Brain Minds Beyond Human Brain MRI project. So the aim of this project is revealing human intelligence, sensitivity, and sociality at brain circuit level for the early detection and the intervention of psychiatric and neurological disorders. So this project collected multi-site brain MRI data of patients with psychiatry and neurological disorders and healthy controls. So we will start to release the quality controlled dataset from more than 7,700 participants from this year. So now we are working hard to finalize our dataset. So this shows the contents of our dataset and the number of patients included in this dataset. So neurodevelopmental disorders, schizophrenia and anxiety disorders, obsessive compulsive disorders, and feeding and eating disorders, disease of nervous system. This includes Alzheimer's disease and Parkinson's disease, and also healthy controls. Okay, so this shows the data flow of our project. Okay, so each site measured MRI data and upload our server, XNAT server. So I am a small user of XNAT in Japan. So, okay, and we conducted data curation in script-based and also manual. Manual means human-based curation. Yeah. So, and we move curated data to distribution server. So in this project, we used Aspera Connect. It is a high-speed network service provided by IBM. Yeah. So, and in parallel, so at RIKEN, our MRI data are pre-processed. And we share curated and pre-processed data for members of our project and also for public. So in Japan, face information included in MRI data are recognized as privacy information. So for public use, we remove all of face information for 7,000 people. So it's very hard work. Okay. And we already published a part of our data set, and we will release more high resolution and measured by advanced protocol from this year. Okay, so as you know, multi-site projects face many difficulties. So one of the most significant difficulty is data governance. Okay, so we have taken 3 actions to preserve data governance in this project. Okay, so one is we developed web application of data governance. So in this app, so we can check the status of the data at the individual level very easily. So for example, data uploaded, curation, and pre-processed in individual levels. Yeah. So it is very important because quality control were conducted in parallel site. Yeah. Not only one same site. So in that case, we should check the status of the data. Yeah. And okay, second is we fix the metadata for dataset. Yeah, it is very messy. Yeah, but yeah, we fix the metadata for catalogs, so it is very important to check the contents of data. Okay, and it is most important point. So we held training course for quality control and data analysis. Yeah, education is very, very important. Yeah. Okay, so one outstanding feature of this project is focusing harmonization. Okay, so we adopted 2 approaches, prospective and statistical harmonization. Okay, so in prospective harmonization, we develop a unified measurement protocol of MRI data acceptable in a clinical site, not only research site, yeah, with finer resolution. Okay, so this shows, this left figure shows the MRI image, measured MRI image of same individual measured in 5 different sites. So, yeah, we try to develop a unified measurement that results in a similar data result. So even in different site, different scanner and different MRI company. Yeah. But of course, we cannot control all of differences arised from different sites and different scanners and different companies. So in that case, statistical harmonization is very important. So, okay, so in this, statistical harmonization, we designed and conducted traveling subject. So traveling subject means each participant was measured in the multiple sites around Japan. So this participant traveled in Japan. So we called traveling subject. Okay, so by minimizing the effect from site differences, we developed a robust biomarker using MRI data, so for measure depression, and that can be generalized to independent data. So this harmonization approach allows the development of AI-based biomarkers with high generalization performance that can withstand clinical application. Okay, so in our project, we collected several types of MRI data, including structure and functional data. So today I will introduce a biomarker developed from resting state functional MRI. Okay, so as some of you may know, resting state fMRI is a functional imaging techniques that capture brain activity during resting state for 5 to 10 minutes. Okay, and so for this modality, we said participants that relax without thinking any particular things. It means resting. Okay, so resting, and so we can get such a time course of MRI signals in each voxels of brain, and we computed a correlation between time course of across different area of brain. And we call it connectivity matrix. And we call this system resting state functional connectivity, FC. Okay, this functional state, resting state functional connectivity is widely used tool for investigating the brain functional network. And several previous studies demonstrated that this includes rich information about individuals, such as fluid intelligence and length of education, and so on. And now, resting state FC is a useful tool for development of biomarker of psychiatric disorders. Okay, so I show the simple diagram of development of brain marker for disease diagnosis. So we collected data from healthy controls and patients. And we computed functional connectivity like this. And we input this data to machine learning algorithm to derive the correlation between functional connectivity data and disease levels. In such case, this is two-class classification problem. So major algorithm is sparse logistic regression or L1 sparse logistic regression, or recently RAS is very sophisticated algorithm for two-class classification. And this two-class classification identify connectivity patterns that characterize disease. This is an example of connectivity patterns in ASD patients. ASD is autistic spectrum disorders. Yeah. In that case, we use sparse logistic regression. Yeah. Okay, we develop several types of diagnostic markers. So ASD and the subtype of depression, it is specific melancholy types of depression, and schizophrenia and the major depressive disorders. So our major depressive disorder has a high generalization performance even with independent data collected overseas. Okay, under our depression diagnosis marker has been approved as first AI-based SAMD software as a major medical devices for psychiatry diagnosis. So it shows more than twice as stable as physician's diagnosis. Yeah. I'm not a medical doctor, so I don't know. So I don't know whether physician's diagnosis is not stable. Yeah. Okay, and it is prospective validation of MRI at multiple sites and vendors. This shows this biomarker show highly generalizable even in different MRI companies and different types of scanners. Okay, so now we release two data sets, including psychiatry and neurological disorders and healthy patients. Okay, so already we released 1,717 samples. So this includes patient and healthy controls and also traveling subject data. So this data set has been downloaded over 5,000 times. And we will release this new data set. So it was measured by more finer resolution and advanced protocols. And this is over 7,000 samples. And for this data set, we used schema standardized protocols of data set. Yeah. Okay, so this shows the status of our data set in worldwide open data set focusing on psychiatry and neurological disorders. So we performed a comprehensive survey of publicly shared databases of psychiatric and neurological disorder worldwide. Target diseases, mood disorders, developmental disorders, schizophrenia, Parkinson's disease, dementia, and eating disorders. So we found over 23,000 samples in public data. You can see almost major samples from neurological disorder, so Alzheimer's disease. And you can see patients with psychiatric disorders very few. Yeah. And this scatter plot shows the number of samples included in dataset and the number of sites included dataset, and the marker size indicates the number of diseases included dataset. So, okay, so this is our already published data, and this is our new data set. So this shows our data set has one of the biggest data set focusing on psychiatry and neurological disorders. Okay, so now we are running new Japanese flagship neuroscience project, Brain Minds 2.0. So, okay, so the aim of this project is promote research to discover new drug diagnostic and therapies for dementia and related disorders, and strengthens international network to maximize research impact and foster collaboration between basic and clinical researches in academia and industry. And we try to develop a digital brain platform using mathematical models. Okay, so we now try to build Brain Mind 2.0 digital brain platform. So we try to connect cross-modality, cross-species databases, such animal database and human databases. And we will develop several frameworks, including cross-species integration space, cross-species search, and one-stop analysis. Okay, so our next step is optimizing systems and technologies we can use. So there are several levels and several phases, so we should choose optimal option in each levels and each phases. For example, metadata systems, there are several systems, DICOM, NIFTY, BITS, and schema.org. And there are several options for database system, XNAT, OpenMinds, BrainInf, Brain Life, and Aspera Shares, and it is a RIKEN CBS repository. And we need find optimal infrastructure for collecting data and publish data. So Aspera Connect is very strong infrastructure. So, but it is very expensive. Yeah. So, and we set distribution server, computing server also. And it is very big challenges to fix the data types we will release. Yeah, even in humans, so there are several types of data. And I'm not animal researcher, so I think this includes several types of data. Yeah, so it is very big challenge. So we welcome any collaboration with you. Yeah. Okay, that's all of my today's talk. Thank you for listening to my talk. And yeah, I show the QR code for our data portal. So if you are interested in using our data set, please visit. Yeah. Thank you.